Thank you, HUD, for inviting me to giving us the chance to present some of our results. So let me first just tell you a little bit about Amicus Therapeutics. Is a company, one of those companies that's focused entirely on developing therapies for rare diseases. In fact, we have multiple programs now in various stages of clinical development. Oops, sorry about that. Multiple stages of clinical development, including programs in Fabry disease, Pompe disease, Gaucher disease, as well as Parkinson's and Alzheimer's. You might not initially think of these as rare disorders, but in fact we're focused on genetic forms, particularly in the case of Alzheimer's disease, early onset familial forms, or in the case of Parkinson's, these are patients who are Gaucher carriers that develop Parkinson's disease. So what I'd like to talk about today, though, is our most advanced program. This is for Fabry disease. This is the development of a pharmacological chaperone, AT1001, or also referred to as Amigal, for the treatment of Fabry disease. So Fabry disease is caused by mutations in the Jolla gene. It is a monogenic X-linked disorder, and there are over, now it's probably already out of date, there's probably closer to 650 different mutations, known mutations, that cause this disease. The Jolla gene encodes the enzyme, the lysosomal enzyme alpha-galactosidase A, and a deficiency of this enzyme leads to the accumulation of the lipid global triacylceramide. This disease affects both males and females. It's not always thought to be the case. It used to be often considered that the females were carriers. This is clearly not the case. There are around 5,000 to 10,000 diagnosed patients worldwide now, and close to half of them are female patients. And over 3,000 of these patients are on ERT. That's the current therapy for enzyme replacement therapy for patients with Fabry disease. Fabry disease can lead to, it has a very broad clinical spectrum, but it can lead to severe episodes of pain, kidney failure, heart disease, stroke, and premature death. And we believe this is something that's interesting that came up earlier in discussion about how many patients actually have missense mutations. As you'll see from our therapy approach, this is very important because you have to have a protein to rescue, and usually these are proteins that are, we're usually looking at proteins that are made from missense mutations. But in this case, this disease could be severely underdiagnosed, like a lot of other rare diseases, because some of the milder mutations, particularly missense mutations, may be either not recognized or often go unrecognized. So there are a few reports that suggest that this disease could actually be as much as 10 times more common than we currently think. So what are pharmacological chaperones? So let me first tell you what pharmacological chaperones are not, because we often get this confusion. They are not molecular chaperones. Molecular chaperones are proteins that often help other proteins to fold. That's not what we're doing here. What we're doing is actually identifying small molecules that that are orally available and selectively bind to enzymes to stabilize them. So as you heard a very detailed talk this morning about how the complex quality control system that cells have within the endoplasmic reticulum that affect cell folding and trafficking, and often these mutations impart instability or cause the proteins to misfold. These proteins can be quickly degraded and never make it to their final location, despite the fact that many missense proteins, if you can get them made, retain some residual activity. So our goal is to identify, as I mentioned, these small molecules that can bind. All our molecules at this point are active site binders. So they bind to the active site of the enzyme, they stabilize it, and this helps the protein then to escape or bypass the quality control system of the endoplasmic reticulum to restore its normal trafficking. We increase levels of the enzyme in the lysosome, and then we have several things working in our favor that allow the dissociation of the molecule so that substrate can be turned over. In this case, one is that our molecules tend to have a higher affinity at a neutral pH of the eye relative to the lysosome. Secondly, the high concentrations of the substrate in the lysosome compete at the active site with the small molecule inhibitors. And finally, we actually control the the concentration or the drug levels. So, for example, all of our drugs will have considerably shorter half-lives in the tissue than they have to, relative to the half-life of the enzyme. So the reason this is important is because, again, these are inhibitors. So if you think of when you dose a drug, you go through normal drug levels of a kind of a pulsatile fashion. When the drug hits high levels, we can favor stabilization and trafficking. When the drug washes out, now you can liberate the enzyme to do its job and turn over substrate. And we'll show you some more of that later. So in this program, this is the molecule. This is our AT1001. One of the first things you'll notice about it is that it's an amino sugar analog of terminal galactose of alpha-galase natural substrate. So it's very similar in structure, the biggest difference being this nitrogen in the ring relative to this oxygen. It's a potent inhibitor. It does bind to the active site. We confirmed this in a collaboration with Dr. Greg Petsko's lab up at Brandeis University. This is the crystal structure showing our molecule here setting in the active site of the enzyme. Now, when this molecule binds to the enzyme, it actually imparts stability to it. We can actually measure that using a thermostability assay. So what you see here is if you take recombinant alpha-galactosidase A at pH 7.4, so this would be like the neutral pH either at the endoplasmic reticulum or when you infuse an enzyme into circulation at the neutral pH, you can see the enzyme, if we add an environmentally sensitive dye like Cipro orange, we can add heat and watch the enzyme actually unfold. So this is a measure of unfolding. And the lower the temperature is, the more unstable that the enzyme unfolds at, the more unstable the enzyme is. So we can unfold this enzyme by adding heat. You can see it's got probably about a TM or a melting temperature of around 45, 46 degrees Celsius. Now, if you take the same enzyme, not surprisingly, these enzymes are lysosomal enzymes. Their optimal pH is an acidic environment. So if you follow the red line here, you take the same enzyme, put it in pH 5.2 and add heat, you can see it's considerably more stable. You have to add another 10 degrees Celsius before you start to see unfolding of the enzyme. Now, one of the things we found is our molecules, because they bind to the active site, they can actually impart stability to the protein. So you can take the same enzyme that was unfolding at pH 7.4, around 45 degrees Celsius, add in our chaperone, AT1001. In this case, it was about 100 micromolar, and you get this big shift in thermal stability at neutral pH. So this does a couple of things for us. One, it tells us partially how our molecules are working. We're binding and stabilizing these proteins, the neutral environment of the endoplasmic reticulum to promote folding and trafficking, normal trafficking of these proteins. You can imagine if you have a missense mutation here, the protein's often even more unstable than the wild-type protein that you see here. But it also, as I'll come back to later on, gives us another way to potentially make some ERTs better, particularly ERTs where the enzymes are particularly unstable at neutral pHs in the plasma. Some enzymes are cleared very quickly or denature very quickly, and you could use this technique to stabilize and increase the half-life of the enzyme itself and its activity in circulation. So we'll come back to that in a little bit. One of the important things to remember, though, are all our molecules, in particular AT1001, it is an inhibitor, but it's a reversible inhibitor. All these molecules have to be reversible, so they can bind once they get to their final location, can drop off. And this is just showing you what I mentioned earlier, is that if you look at the inhibition curves and you determine the KIs at the acidic pH of the lysosome and compare it to the neutral, more neutral pH, similar to the endoplasmic reticulum, our molecules are about threefold more potent at the neutral pH. So it works in our favor in releasing that enzyme once it gets to its final location of the acidic lysosome. Now, the first thing I'm going to tell you about chaperones that are counterintuitive is that if you take this inhibitor and you treat cells for 4 to 5 days, and these are patient fibroblasts from a male Fabry patient with the R301Q mutation in alpha-galactosidase A, and then take the lysate and measure the levels of the protein as a function of activity, you actually see increased activity. So I should caution you, though, this is really a function of levels here. We're not measuring activity because strictly in situ or in vivo activity because you've diluted out your inhibitor by looking at the cell lysate. So this raises a couple of questions. First of all, can we do this? Can we see the same type of increase in vivo? And secondly, can we translate this increase in enzyme levels into increased activity? So to test this, we used a transgenic Fabry mouse model. This is a mouse model that has a knockout alpha-galate background, but it expresses the R301Q, the same mutation we just showed you, GLA. And if we treated these mice for 28 days with AT1001, in this case they were orally administered, it was just put in the drinking water, and they're allowed to drink ad lib for 28 days, and then measure enzyme activity in 3 of the relevant tissues for Fabry disease, the skin, heart, and kidney. You can see similar increases in these light blue bars in enzyme activity. Now, does this, though, translate this? I'm calling this activity, but in reality, again, this is a lysate. We're taking tissue, we're grinding it up, measuring activity, diluting out the inhibitor. So we definitely have increased levels, and this correlates with Western blots, but do we have increased activity? And that we can measure by looking at the substrate. If we have increased activity, the enzyme substrate levels should drop. And that's what you see, actually, if you look at all 3 tissues. The dark blue bars on the right, with treatment, we've significantly dropped the levels of substrate. So we have, even with daily dosing for 28 days, we're able to significantly drop levels. In fact, if you treat these mice for an even longer period of 24 weeks, we can drop levels of substrate even further. But this brings up a second counterintuitive point of chaperones, which actually makes sense if we're thinking of these inhibitors, but what if we dose less often? Usually you give more drug to get more of an effect, but could we get a greater effect by giving less drug? And the rationale here comes back to it being an inhibitor. These enzymes have, even the mutant enzymes, have relatively long half-lives in the lysosome on the orders of days. These molecules have half-lives on the order of hours. So we can get a net gain. So instead of dosing daily, what if we dosed every other day? The drug would wash out, it's gone within the first day. The second day, you still have elevated enzyme levels. You should be able to turn over even more substrate in the absence of the inhibitor and see a greater effect. And in fact, that's exactly what we see. So if we repeated the same study, and this time we dosed by oral gavage, and you can see in the first 2 bars, difference between the treated and the animals treated daily, and we saw the same type of decrease in substrate levels. Now, if you treat every other day in this 3rd bar, you can see we decrease substrate levels even further. In fact, this is the rationale that we use for the same molecule in our phase 2 and phase 3 clinical trials. We actually dose patients every other day in an intermittent dosing manner. The other thing that you can also see here was we also included an arm where we infused or injected Fabrazyme once a week for 4 weeks, and you can see the type of substrate reduction we're seeing in these animals is very similar to what we observed with Fabrazyme. The next question, this was all for 1 mutation. I mentioned there's over 650 mutations in Fabry disease. Do we have to test every mutation to determine if there are responses? Well, yes, for the most part. So we already know that out of the 650 mutations, there's probably about 200 that are either nonsense or there are mutations located in the active site that basically destroy the activity of the enzyme, and we're not going to rescue those proteins. But for the other 450, we created cDNA constructs for all 450. These were all missense mutations or small in-frame or insertion deletion mutations. We expressed the mutant forms transiently in HEC293 cells and then incubated for 4 to 5 days with our drug from a concentration of 17 nanomolar to 1 millimolar. Then monitor activity either using a synthetic fluorogenic substrate or Western blot, and use this then to determine 2 things. One is we want to understand the magnitude of the increase in activity, and we can compare this, of course, to wild type to get some idea of where we end up. And we also importantly need to know the EC50, at which dose, because the mutations in the protein also affect its potency. Some mutations will lower the potency of the drug. And so one of the things that's important for us is to understand we may see an effect, but can we see an effect at a clinically achievable dose? This next slide looks very complicated because there's a lot of data on here, but we tested all these mutations. What you see in red here is the activity for each of the mutants prior to treatment. In blue is after treatment, the maximum activity that we saw at any of the doses that we tested. And you can see as these are laid out along all 7 exons that we do have responsive mutations in all 7 exons, and a lot of the mutations actually do in fact respond to our drug. Here are the EC50s, and if you compare the most potent to the least potent, there's about a 1,000-fold difference in its EC50s. So it's very important for us to understand this, to correlate this together with our PK data in humans. So now we have all the tools we need to identify the patients that we would expect to respond. Do they respond at the particular dose level we expect to achieve when administering dose X? So with this data in hand, and together with some of the other preclinical safety results, we found that, for example, we had a very acceptable toxicity profile when we tested AT1001 at high levels in rats, dogs, and monkeys. One of the things we did observe was male infertility in rats, and this was reversible. So I'm just going to touch on a few highlights from our phase 1 clinical trial results for AT1001. Basically, there were three major studies: a single ascending dose, where we looked from 25 to 2,000 milligram dose, a multiple ascending dose, and a single dose where we looked at for QT elevation. In all of the studies, AT1001 was generally safe and well tolerated. And the particular point is worth making is in the multiple ascending dose, we found that AT1001 led to a statistically significant dose-related increase in alpha-gal A activity in white blood cells of healthy volunteers. Or these are not Fabry individuals with Fabry disease. These are healthy individual volunteers. The reason you can increase this, turns out we had about a twofold increase, is because even wild-type proteins, we find many of them don't completely 100% efficiently fold in traffic. And it varies from protein to protein, but we've now found that a lot of wild-type proteins, you can increase their efficiency of trafficking and folding. And this is exactly what we see here. This is relevant, I'll come back to it a little bit later, because when you look at females, I think I forgot to mention earlier that females actually, because it's an X-linked disorder and there's X inactivation, their tissues are actually made up of a mosaic of healthy and diseased cells, cells that are either expressing the wild-type enzyme or the mutant enzyme. And so all females already have 50% levels if you just take some tissue and measure activity, and our chaperone will double that level, but that is not necessarily double. We were not unable to separate out the effects on the mutant and disease cells versus the wild-type cells, unless we look at substrate levels, and we'll come back to that. So we moved on to a phase 2 clinical trial, and I'm not going to go through all these protocols in detail, but I just wanted to put this up there to show you there's really two reasons for doing a phase 2 trial. Obviously, the primary reason is to demonstrate safety in patients, but it's also an experiment. It's an experiment that allows you to helpfully identify a dose, a dosing regimen, your patient population, the appropriate endpoints, and the number of patients that you need to properly power a phase 3 study. And so that's why you'll see here, if you look across the 4 different studies that we conducted, various doses. Some of these are escalating doses. Some patients had kidney biopsies, others didn't. There's both males and females, depending on the study, and even the durations of the study are different. What's not listed here is that we also have a long-term 205 study, which is a long-term extension study. So many of the patients are continuing in this extension study. In fact, we now have data on about 22 patients who have been on AT1001 for over 3 years, and 7 of those patients have been on AT1001, been taking AT1001 for over 4 years. So this gives us a cumulative of about 85 patient years of data with AT1001. To date, in all of our phase 2 and extension studies, we've experienced 23 serious adverse events. All of them were deemed to be unlikely or unrelated to AT1001, so I won't go through all the details. Of most of the adverse events, most common ones were joint pain, headache, diarrhea, all symptoms commonly associated with Fabry disease. And then there were 4 AEs that were considered probably related to study drug. 3 of them were considered mild and were fatigue or upper GI issues, and in all 3 cases spontaneously resolved. And in a 4th case, it was an individual, when their dose was escalated to 500 milligrams, they experienced severe joint pain, muscle pain, tremors, confusion. In this case, the study drug was interrupted, the AEs resolved, and the subject resumed at 250 milligram dose. So here are some of the white blood cell data. So one of the things we do is look at, just like in the phase 1, we look at the activity levels in white blood cells. And these are various time points at different groups, patients that where we looked at their white blood cell either at activity levels either at 12, 24, or 48 weeks. These are different groups. We've kind of combined all the results together here. So it's a little bit, you see a lot of, don't know, wouldn't want to call this noise, but variability here, because remember, some of these patients, first of all, they all have different mutations, but secondly, they also are on different, receiving different doses, or in some cases, different dosing regimens. We've also taken the male patients and divided them into two groups. 14 of the males are considered responders that fit into these groups. You saw shows some kind of statistical significant increase in their enzyme activity levels. And then we had a group of about 3 males that were non-responders. And one thing I'll mention here is that these responders and non-responders correlated very well with what we would predict in our in vitro assay, where we could actually test each mutation individually for response at the drug levels we would expect to reach in these patients. The other thing we needed to look at is for the enzyme replacement therapy for Fabrzyme was a conditional approval was based on clearance of the kidney interstitial capillary GL3. So we needed to look at this as well. This will be our same endpoint that we'll have for our phase 3 trial. So basically, we took kidney biopsies from some of the male and female patients. They were processed in the slides and then assessed in a blinded manner using light microscopy by 2 independent pathologists. For each biopsy sample, we counted 50 to 180 capillaries for each sample were assessed for the number of GL3 inclusions, and you can kind of see an example here. This is one of these cells that has about 9 inclusions. Here's one that's completely clear of inclusion. So it's relatively easy to count these inclusions, and so it gives us a very quantitative measure. And then we can determine the kind of the average number of inclusions per cell for a patient. So if you look at if we look at this as a function of substrate reduction, if you look at this is the results for the kidney interstitial capillary GL3 levels from the kidney biopsies. Now I'm just showing you here, these are the patients, again, the same ones I showed you before. These were the patients who had responsive mutations. We saw 5 out of 9 of the patients had 5 out of the 9 patients who had biopsies at week 12 or week 24 showed a 50% or more reduction of their interstitial capillary GL3 levels. Now, if you look at the patients in during the extension at the 48 who have been on drug now for 48 weeks, all of these patients were receiving the same, at least during the extension, receiving the same dose and dosing regimen, the 150 milligrams every other day. In this case, 7 of the 10 patients had at least 50% or more reduction of GL3 levels in the capillary. Another surrogate, and this is a surrogate endpoint, I should mention. A second potential surrogate endpoint we looked at was urine levels of GL3. And again, if you look at all of the responders, in this case we saw about a 34% reduction in urine GL3. Now, if you only look at the individuals that were in the extension study that were on 150 milligrams every other day, we saw that this even a better reduction in urine GL3 levels. And this is just showing you those patients who didn't respond, the non-responders, they in fact showed elevated urine GL3 as you might expect. So in conclusion from we were able to conclude a few things from this study, including that AT1001 was generally well tolerated. Treatment with AT1001 increased the levels of alpha-Gal A in patients with mutations. This is important that are eligible for our phase 3 clinical trial. The treatment of AT1001 also decreased levels of kidney GL3 in these same good responders as measured by urine or kidney biopsy. We've identified our patient population for our phase 3 study. They will be patients who we would predict to respond at the 150 milligram every other day dose. I didn't talk about renal function, but our preliminary assessment of renal functions, it's not definitive, but it certainly was encouraging. It looks like we see at least stabilization of GFR. And this is similar to, this is what's been observed for ERT, just a stabilization of GFR. Interestingly, what we did also see were trends in decreased proteinuria, and this was nice because we've seen these trends continue in our long-term phase 2 extension study. And this is very promising for several reasons. One is that ERT didn't show much effect on proteinuria. Additionally, patients who have significantly elevated levels of proteinuria tend to not respond as well to ERT. So with these results in hand, we've now, last year we started enrolling for our phase 3 study. We plan to enroll a total of 60 patients in a 6-month trial. This will be AT1001 versus placebo, with our primary endpoint being a demonstration of a 50% or greater reduction in the kidney interstitial capillary GL3, as I showed you from the phase 2 study. And one of the things we're doing to enrich for our patient population is we're obviously using our assays to identify mutations we would expect to respond. And additionally, we're using the GL3 urine assay to enrich for patients who have elevated levels of GL3. In fact, if these patients are too mild, for example, their baseline levels in the kidney of GL3 can be very low, and then demonstrating a drug effect is very difficult. So we need to make sure that we have, so we're using this assay to identify those patients who have elevated GL3 levels. And our studies will includes both males and females. So just a bit of a status update. As of January, at the beginning of this year, we had 36 sites were initiated globally. We've enrolled the majority of the patients, and we expect top-line results by the end of this year. So I'd like to touch on just one more point before I finish here. As we expect, based on our in vitro assays, that monotherapy could potentially be beneficial for about half of the Fabry patient population. What about the other half, the half that won't respond or have nonsense mutations, et cetera? So one of the things we've been working on is, as I alluded to earlier, is that these chaperones could also potentially make ERT better. And here, if you watch Faberzyme, for example, it's relatively unstable at pH 7.4. This is the pH in circulation and at 37 degrees Celsius. And if you just put it in a buffer at this pH and temperature and watch what happens to activity, you can see over the first 48 hours, it drops pretty dramatically. If we add in, together with the Faberzyme, our AT1001 at a clinically achievable dose of 1 micromolar, you can see you can significantly reduce the amount of activity lost as a result of denaturing of the enzyme at this pH and temperature. So can we, so the next question is, can we increase the half-life of Faberzyme in circulation? And so to do this, we took rats and basically treated them with AT1001, 3 milligrams per kilogram of AT1001, administered by gavage about 30 minutes prior to IV administration of a 10 milligram per kilogram dose of recombinant alpha-galactoside assay. And what you see here, either measured by activity or measured by protein levels, is that in fact we do increase the half-life in circulation from about 23 hours to about 58. It's pretty significant. Next question is, does this translate, though, into uptake and increased activity? So return to our alpha-galactosidase mouse model, or Fabry mouse model, but in this case we used a knockout mouse model. So this mouse model has alpha-galactosidase A knocked out, or GLA knocked out. It should have very little residual activity, as you can see here. And then we infuse inject the mice with a single dose of Faberzyme, and sacrifice the mice a week later and measure, in this case, enzyme activity in the kidney. And so you can see, even a week after the infusion, you still see an increase in enzyme activity, as you would expect in the kidney. Then we do the same by administering AT1001 by gavage 30 minutes prior. In this case, the doses were 30, 100, or 30 milligrams per kilogram. And you see a nice dose-dependent increase in enzyme activity in the kidney tissue, suggesting that we've significantly increased the uptake of the enzyme. Now, does this translate into activity? So again, if you look at these mice in the kidney, they have significantly elevated substrate levels of GL3. With just the infusion of the Faberzyme, you can half that. You significantly drop the levels of GL3. And if you, again, when we added the chaperone 30 minutes prior to the infusion, you see a significant, a further drop the substrate levels at all 3 doses. So as a result of any of these preclinical trial results, we have also recently initiated a phase 2 clinical trial where we've just begun enrolling for, it's basically a PK and uptake study where we'll be co-administering ERT, Faberzyme, or Repligal, together with AT1001, and then following the PK of Faberzyme together with uptake of the enzyme. In this case, we'll take skin biopsies, and this is the first study that we've just recently initiated. So in summary, AT1001, or Amicus, physically stabilizes alpha-Gal A, leading to an increase in alpha-Gal A activity in culture cells and shown in vivo. About half of the mutant forms of alpha-Gal A that we've tested respond to or about half of the mutant forms of alpha-Gal A respond to AT1001. So in our phase 1 and 2 trial results, we found that AT1001 was generally well tolerated. Treatment with AT1001 increased levels of alpha-Gal A activity and decreased levels of kidney GL3 in patients that have mutations which are eligible for our phase 3 clinical trials that are currently ongoing. We also have evidence of stabilization of GFR that continues into our long-term extension study, which was also comparable to what's been observed for ERT. The trends of reduced proteinuria have also been observed and are continued to be observed in our long-term phase 2 extension study. We expect phase 3 results for AT1001 by the end of this year. And as I also just mentioned, we just began enrollment for our first phase 2 ERT AT1001 co-administration study. So I would just like to end by thanking, obviously, this work. I feel privileged to have the opportunity to stand up here and present work that has been conducted by a lot of my colleagues back in Amicus Therapeutics over the last 9 years, together with a lot of academic researchers and, of course, the patient organizations who have all along the way we've consulted with, and have also many of the patients who, of course, volunteer for our trials. We couldn't do any of these studies without any of them. So many thanks to everyone that's been involved.
I really like your preloading slide where you did dose escalation. The 30-minute you chose initially, was that based just arbitrary, or did you have to do initial?
30 minutes for pre-administration of the chaperone to the. It's based on our PK studies where we get maximum drug levels.
That was Cmax at that point.
Right? It's around Cmax. Yeah.
Yeah, because it really saturates pretty much from 100 to 300 looks identical at that point.
Yeah.
So that's really neat. And then when you went higher, you looked at activity at 300. If you believe that you got too much residual inhibitors going back.
Yeah, you might. I mean, it's hard to say because these studies were taken, of course, the mice were sacrificed a week after administration, so certainly the drug should be gone by that point. But it's still you're still, especially in the substrate, looking at a function of the activity over that entire time. So if there's a little more inhibition up front, that might have affected the end value. So yeah, you could go too high.
Good question. So when you co-inject, what's the half-life in the serum, and is there? Have you ever even tried pre-binding and then?
So basically combining them and put them. No, we haven't tried that. No, we've only done them partially because we were supporting. This was meant to support the study, the type of study that we would do in the clinic, which would be co-administration of the drug separate. But certainly there could be opportunities to co-administer them ahead of time. And actually some potential real advantages there. Some of these it's been shown that some enzyme replacement, you can actually see them. I think in Pompeii is a good example where myozyme will begin to precipitate in the infusion bag. So if you could stabilize that and prevent that from happening, you could potentially use
Question to actually make it a co-formulation at the start. Cost of manufacture of that would be very.
And there's many things from the point of not only from stabilizing in the bag, you can go back even further into manufacturing the enzyme itself.
Well, the high concentrated sugar itself could be a stable protein.
Exactly.
Okay. Thank you very much.